All right, so video number 2 from my nutrition class. This is a nutrition class. This is not a biochemistry class. Having said that, though, the anatomy of so proteins are made up of amino acids. Now, amino acids come in different shapes and sizes. The reason why I should teach you this is because it all comes back down to how do we track amino acids in the diet. If you're worried about your protein intake, you understand, okay, proteins are made up of amino acids, and amino acids have an interesting configuration. So here's the most basic structure. It's got the center carbon. You don't need to know chemistry. It's got this thing on the side we call an acid group. It's got the element hydrogen. And then this is called an amine group. So amino acid. Notice, wiggling over to the left side, the N is for nitrogen. That is not an atom or an element you've seen in our class yet. And when all things are said and done, and often more is said than done, that's a great line, the vast majority of nitrogen on your dinner plate is in an amino acid. Yes, there's nitrogen in some other molecules like vitamins, but the overwhelming vast amount of nitrogen on the plate is in amino acids. So when it comes to tracking protein in the diet, you could track these fancy molecules using fancy chemistry, or you could just track the nitrogen, assuming there's a certain amount of nitrogen in a gram of protein, and then how much nitrogen's coming into the body, and then fortunately, in the urine, you can find nitrogen, and you can back calculate how much nitrogen in the urine equals protein, and you can just track nitrogen to figure out protein coming in and protein leaving the body. That's the beauty of amino acids, the nitrogen. It's a measurable thing in biology. So let's move my head again. Here they are. You will take a class if you're a nursing student or a science student, and you'll have to learn the name. And if it's a upper division class, you'll have to learn how to draw these. And I did once, and I don't remember how to draw these now. These are all different kinds. Some of these have sulfur, the element sulfur. Some are acidic, some are basic, some like fat, some like water. This isn't the sugars. They all kind of look the same. They all kind of act the same. The building blocks of protein are 20, and they're extremely different from each other. That's the key. And then I said that some are essential. So some of these you have to eat, these different kind of molecules, and some you don't. And we know that for humans, 9 amino acids we cannot make, or we can't make in great amounts. And 11 of them we can. So if you had to be the cheapest the cheapest dietitian for NASA on Earth, and you're like, look, we're only going to send, astronauts to Mars with supplements, you could give 9 bottles of amino acids for the ship, for the spaceship ride to Mars. You're not going to do that, but you could. And the other 11, oh, don't worry, they'll make it. That's really not how we eat, obviously. We can take these total in 20, we can link them together, and we can start making things. This is insulin. Insulin's got 52 amino acids. There's actually a piece of this that's missing. When insulin is made, it's one big, long, giant chain. It's snipped, it's rearranged, and it has a certain shape. Usually when you make proteins in the body, you start with this long sequence. It's called a sequence. And each position can have a different amino acid. So I'm not a mathematician, but that means it's 20 in the first place. Let's say it's 1,000 long of amino acids, this long chain. 20 can be the first one, 20 can be the second one, and you start doing the math, it's an infinite combination. And so you make these inside cells that have DNA. There's a whole process of making these long sequences. They start to fold, they become a shape. This is hemoglobin with 4 different subunits. This is actually hexokinase. It's an enzyme. This image right here is a hexokinase. It's an enzyme that could take glucose and convert it to galactose. It's that kind of enzyme. Remember I said when you consume fructose, galactose, glucose, in the liver, galactose and fructose are converted to glucose. One of the enzymes that does that is hexokinase. Look how incredible that is. Biologic origami. It's amazing when you really look at it and think we do this with every protein we make in the body. A specific shape based on the sequence of the amino acids and which amino acid is in which position. So what if your DNA has the wrong information? What if you have a mutation inside the DNA of your body and in the bone marrow where you're going to make your hemoglobin that goes inside red blood cells, hemoglobin carries oxygen, right? What if you made a hemoglobin that was slightly wrong? Now, I believe hemoglobin is 574 amino acids long, the molecule hemoglobin. So here it is. There's 4 of them together. It becomes a giant, like, combined molecule. I think that's the heme group with the iron, and then oxygen sits right in the middle. If one amino acid is wrong, the shape is slightly different. You may not be able to tell. I wouldn't be able to tell, but the body knows. And that actually ends up changing the shape of the entire red blood cell and the ability of the red blood cell to carry oxygen. Fascinating. None of us look the same. Other than twins, none of us look the same. How is it that we're all using the same kind of proteins, yet we all look kind of different? Part of that is, is genetic arrangement. Part of that is epigenetics. Part of that is we all have similar proteins, which can be slightly different, but as long as they work, even though they're different, we're good to go. With hemoglobin, you can probably make a mistake in another part of the hemoglobin, and maybe there is no sickle cell anemia. But if you swap out the wrong amino acid at a specific position, it can have dire consequences. That's sickle cell anemia. Okay, we're going to end on that.